life cycle of angiosperm. In this life cycle, we are going to learn how a reproductive structure called flower from one generation able to reproduce once again a new flower. Here I am going to explain this in 8 different words. Simply I am going to explain that as microsporogenesis, megasporogenesis, fertilization, embryogenesis, endosperm formation, seed formation, fruit formation, germination. So 1, 2, 3, 4, 5, 6, 7, 8. Not all the events are major events here. Now let me explain the first one, microsporogenesis. So microsporogenesis. Here the process consists of a male part called androecium. This androecium consists of stamen. Stamen consists of 2 important parts called filament and anther. Anther is a bilobed structure which consists of, each lobe consists of 2 compartments, so 4 compartments. Each compartment consists of pollen sac. The pollen sac contain microsporangia. This is 2n in nature, that is called diploid. This microsporangia, in that a very important cell is called microspore mother cell. They are all in 2n status. That microspore mother cell will undergo cell division called meiosis, reduction division, and they are able to produce 4 microspore that we can call it as a microspore tetrad. Let me consider one microspore in that. This one microspore later undergo with the mitotic division, mitosis. Once they undergo mitosis, they will enter into the 2-cell pollen grain. In that you can find vegetative and generative cells. Once again, the generative cell will undergo mitosis and they form 2 male gametes. This vegetative cell undergo mitosis to form pollen tube. Now let me move on to the next process, that is in the female part, gynoecium. Gynoecium consists of a lot of carpels that is grouped together to form a functional unit called a pistil. This pistil is a functional part where you can find 3 important parts: style, stigma and ovary. Inside the ovary, along with the placenta, you can find the ovule. Ovule, it is called as Megasporogenesis, which is 2n in nature. from that functional cells called megaspore mother cell, which is also 2 in nature diploid. This will undergo meiosis to form 4 megaspore. Out of this, 3 will be degenerated and 1 will be available for next process. So 1 functional megaspore. The 1 functional megaspore undergo with a mitotic division, mitosis. Actually, this megaspores are n in nature. Here also the microspores are n in nature. This will undergo 3 repeated mitosis and will form very important thing called a embryo sac. In that, we able to find one end called a micropylary end, another one is central or middle part, the third one is chalosal end. In micropylary end, you can find 3 cells, and central you can find 1 cell, and chalosal you can find 3 cells. See, this cell alone contain 2 nuclei, so we will call this as a polar nuclei. And this micropylary end will have 1 egg cell and 2 synergids. This polar sac will be defined as 7-celled 8 nuclei, because except the central cell, all are having only 1 nucleus. Now, very interestingly, this egg cell is waiting for the male gametes, the 2 male gametes which is going to come and fuse. So they are waiting for the 2 male gametes. Out of this 2 male gamete, 1 gamete will fuse with the 1 egg cell, the other male gamete will fuse with the polar nuclei. So let me discuss the third important thing is called fertilization. During the process of fertilization, 2 processes, syngamy and triple fusion, will take place. The polar nuclei combining with 1 male gamete is called triple fusion. The egg cell combining with the sperm cell, that means male gamete, it is called syngamy. Together it is called double fertilization. After the double fertilization takes place, we can find 2 things, zygote and primary endospermic nucleus. These will turn into endosperm, which is literally works as a nutrition, and zygote will be turned into a proembryo, globular embryo, heart-shaped embryo, mature embryo. Finally, this is ready to develop into a 2 important thing is called embryonic axis. The other one is cotyledon. This is literally going to get packed. That means this embryonical axis, cotyledon, endosperm, all are packed as a seed. Along with the ovule, seed coat, and ovary will form a fruit and fruit wall. So this is the formation of seed and fruit, as I already explained this process. So you can find seed and fruit formation takes place, and finally the seed or fruit, if it is eaten or maybe directly the seed is used, the seed will be germinated. During the germination, the embryonical axis of the seed will be turned into root, shoot. Shoot will form flower, and this flower is what I mentioned as flower to flower. Flower to flower. So in first generation, the flower 1, using its process called Microsporogenesis and Megasporogenesis, okay, using Megasporogenesis, it able to form microspore and megaspore. First the pollen grains are developed, and then pollen grains enter into the stigma, and they create the pollen tube and reach the ovule. Inside the ovary, inside the ovule, this megasporogenesis takes place, and they form egg cell, and this pollen tube carry the two male gamete and fuse that and form syngamy and triple fusion, and it becomes a double fertilization. Later the syngamy will give rise the zygote, and triple fertilization will give rise the endosperm. These zygote turn into a embryo, and embryo will be turned into these are all happening by a process called embryogenesis. So microsporogenesis, megasporogenesis, fertilization, embryogenesis, endosperm formation, seed formation, fruit formation. Finally, this is seed is facing the soil, where it is triggered and converted into a root, shoot. From that shoot, the flower formation takes place. So this is a wonderful sequential process that our nature is doing for us. So let me enjoy it. Thank you.